being recorded and will be made available on the Remplex webpage. As a reminder, we request that you please keep yourself muted during the presentation and for the Q&A session after the presentation. My name is Melissa Perdue. I'm with Pacific Northwest National Laboratory, and I'll be moderating today's seminar on behalf of the Center for the Remediation of Complex Sites, called Remplex. Remplex is a PNNL platform that couples unique core capabilities and expertise with state-of-the-art facilities and physical assets to develop, mature, and deploy advanced technologies to solve complex issues of contaminated subsurface environments. Today's seminar by Tom Brouns, Inci Demirkanli, Vicky Freedman of PNNL will discuss how our capabilities are integrated for the remediation of complex sites. Tom Brouns manages PNNL's environmental management sector. He has more than 30 years of experience developing and managing research, development, and demonstration projects and programs involving government, industry, research, and university partners. Mr. Brouns is a chemical engineer with extensive technical and management experience in the disposition and remediation of chemical and nuclear waste, including processing, disposal, and subsurface cleanup. Inci Demirkanli is a senior computational hydrogeologist at PNNL with a background in subsurface flow transport modeling with applications in remediation research and subsurface energy storage systems. Inci primarily manages projects providing technical and programmatic support to DOE's Hanford site for characterization and monitoring of contaminants, assessment and implementation of remediation technologies, and remedy impacts and optimization. Vicky Freedman is the Soil and Groundwater Program Manager for the Environmental Management Sector at PNNL. Dr. Freedman leads the Deep Vados Zone program, which leverages investments from basic science, applied research, and site contractors to address the complex region of the Deep Vados Zone at Hanford. Her research has focused on numerical studies of coupled hydrodynamics, contaminant transport, and geochemistry in environmental systems since she joined PNNL in 2000. During and after the presentation, I encourage you to submit any questions you have via the chat box, which will be monitored, and the questions will be passed along to Tom, Vicky, and Inci for discussion. I'll be posting a link in the chat box to a Remplex survey where we invite you to provide feedback on today's seminar and offer recommendations for future topics. With that, I'll turn it over to Tom to kick us off. Tom, the floor is yours.
Thank you, Melissa, and thanks everyone for joining us today. I am going to kick off this talk. I think most of the presentation is going to be from Vicky and Inci, but I wanted to get us kicked off, put some framework around what we mean about complex sites. And so with that, let me just move forward. So the agenda today, I'm going to kick it off with discussing kind of the challenge. What is a complex site? What are the distinguishing features of complex sites? Then Inci will take over and discuss kind of key capabilities that are important for complex site remediation, including the characterization, feasibility investigation, remedial option investigation, and all the way through implementation, those capabilities. And then Vicky's going to join and discuss really a specific example of how those key capabilities are integrated in the example of iodine-129 remedy selection support that PNNL has been involved in, and Vicky's been leading that effort. So that's the outline of today. Let me kick it off with just a few introductory comments about complex sites. So complex sites really is a recognition that there is a large number of remediation sites in the US, estimated at around 300,000 sites, that are technically complex, that the complexity of those sites limits their the ability to remediate and reach closure or reach the desired end states in a reasonable time frame. EPA's analysis in 2004 introduced this notion of complex sites. The National Academy of Sciences National Research Council in a 2013 report highlighted that technical complexities are really limiting the ability, preventing the ability to close sites for many, many decades. And it was that complexity driven by extensive contamination, heterogeneous geology, large releases, source terms, their distribution, that really was driving these long timeframes for reaching an appropriate cleanup end state. And subsequent to that, ITRC issued guidance around complex sites in 2017 that really focused on this challenge of complex sites where the remediation progress is uncertain, where the time again to closure it was very long and not within a reasonable time frame that require really greater attention and different approaches to handling these complex sites. It's also a large budget problem, as this time for cleanup extends many decades for some of these sites. So what makes a complex site? Well, it's a number of things, and I'm just going to highlight a few here to get everyone kind of oriented to what we're talking about. One scenario is there are multiple sources, multiple contaminants in the subsurface. In this example shown, tanks and waste discharge sites, for example, at Hanford, commingling in the subsurface and creating additional challenges for remediation. Another aspect of that is shown a little bit differently in this graphic, is that the environment and the geology, the heterogeneity in the subsurface is very complex and drives a number of processes, perched water zones, capillary breaks, lateral spreading, faster or slower paths to groundwater from contamination in the vadose zone are all adding to the complexity of the site based on the geology. Another aspect is shown here is in this graphic is kind of pointing to the many processes involved in contaminant fate and transport. The fact that there are physical processes and chemical processes occurring in the subsurface, or that can occur, with some contaminants and in certain geologies, all of these processes occur and are critical to the understanding the fate and transport of the contaminant. So it's that impact of these multiple processes on fate and transport that add to the complexity. And the last one I want to mention is really about the process that certainly in the United States we follow for remediation, both for characterizing, assessing the contamination, and ultimately selecting a remediation strategy and implementing that remedial action through either the RCRA or CERCLA process. From a regulatory standpoint, that is historically has been a fairly linear process, but in reality, it requires for complex sites a much more adaptable decision process and progress of cleanup. You may start with a corrective action, pump and treat to try to reduce source contamination or source control, and ultimately that is not going to get you to the end state. So the adaptive management methods are critical for achieving the end states, where you refine your conceptual model and look at how to reach an end state in a reasonable time frame or within reasonable cost through adaptive management. So a third aspect of this, or another aspect of this complexity in this process, is that for certain areas, maybe the regulatory framework is such that you have certain regulators owning responsibility for one part of the problem and other regulators in control or in review and approval of different aspects of a contaminant plume that extends over different operable units or different regions of a site. So that regulatory complexity can also be a factor in the overall nature of this long time frame, difficult to remediate site. So with that, that kind of sets the stage for what we mean by the complexity, and I'll turn it over to Inci, who will proceed with talking about the core capabilities needed to tackle this type of problem. Inci, you should have control.
Thanks, Tom. So let me take the control and advance the slide. Great. So I'll start with giving you a summary of our core capabilities. PNNL stewards scientific and technical capabilities in 4 core capability areas, supporting multiple DOE offices, NRC, EPA, and some other sponsors. These capability areas align with different phases of the circular process that Tom was just describing. Under the nature and extend area, we focus on site characterization activities, addressing data gaps in conceptual site models for the complex sites. We utilize our characterization capabilities in order to investigate distribution, fate, and transport of contaminants in these areas. This information provides a technical framework for remedial design and also remedial action as well. Our remedial design capabilities focus on evaluating remediation technologies from all the way from bench scale to pilot scale as the basis for remedial actions. This involves quantification of contaminant flux and attenuation mechanisms and identification of controlling mechanisms in the subsurface as well. PNNL also has significant experience in transitioning remediation technologies to field-scaled applications in the remedial action area. This transitioning requires very close collaboration with the cleanup contractors. Our expertise in this area involves remediation systems engineering, flow and transport assessments, and the use of natural attenuation mechanisms. And finally, we use our experience in risk assessment and environmental remediation to support the development of frameworks for evaluating risk-based remediation endpoints. Development of such a framework for an appropriate end state requires structures and systems-based technical approach consistent with regulatory requirements. This is where we also focus on technical approaches to support adaptive site management strategies. So overall, these capabilities are critical for developing successful remediation strategies, especially for complex sites. So these core capabilities that I just summarized, they create the basis for Remplex as a shared forum. Through collaboration facilitated within the Remplex framework, we can leverage our existing capabilities for supporting remedy decisions for complex sites. So Remplex is intended to provide this framework for expert knowledge, technology, and data exchange. Within the Remplex framework, we have 6 core elements, and I'll just try to briefly describe these. So we start with the development of conceptual site models. This is for establishing technical basis for remedy approaches at a site, especially for complex sites. And providing technical basis for adaptive management is another focus of Remplex framework. This is for developing remedial approaches that would adapt to evolving site understanding over time. Synergistic remedial approaches is also another important element. We would be looking at integration of different remedy approaches in combination to enhance the mitigation of contaminants in the subsurface. We have 2 monitoring focus areas within the Remplex framework. These are risk indicator monitoring and adaptive predictive monitoring. Risk indicator monitoring has this long-term look. It would focus on long-term monitoring at complex sites for developing exit strategies and implementing these strategies. And adaptive predictive monitoring has a focus of reducing the uncertainty associated with complex subsurface processes at a given time and location at these sites. And finally, national, international guidance development is another important element of our Remplex framework in order to provide our feedback, technical understanding, and make it available to larger community. So our capabilities associated with these elements, all of these elements, create an integrated framework for developing a successful remediation approach at a complex site. But for today's presentation, we will primarily focus on the first element of this framework, which is the development of quantitative conceptual site model, QCSM. However, it is important to note that all these capabilities are interrelated, and some aspects of today's discussion will also be related to other elements of Remplex framework as well. And also within the Remplex seminar series, there will be some subsequent seminars to provide more detailed discussion of our capabilities associated with these other elements. So what is a QCSM, quantitative conceptual site model? Conceptual site model is a collection of site information gathered primarily from site characterization activities. It is developed to provide a conceptual understanding of the site, to evaluate the potential risks to human health and also the environment in order to support remedy decisions. A CSM is considered a living representation of a site, which means it is developed during the site characterization activities, early stages of the regular circular process. But it is also continued to be refined iteratively after remedies selected at a site and when it is being implemented. The development of a QCSM, one, provides technical foundation to inform remedy approaches and associated timeframes. And two, it targets developing actionable information on contaminant distribution and its interactions with the subsurface environment. The quantitative nature of conceptual site model is typically derived from supporting analyses and modeling activities associated with the characterization data. All right. Characterization of contaminant mobility and its behavior is the key element for developing a QCSM, because these drive the risk in vadose zone and also for groundwater at complex sites. Through characterization activities, we try to understand contaminant distribution and attenuation mechanisms, and we also try to quantify the specific flux into groundwater. So our main goals, overall goals for characterization activities at a complex site include evaluation of risk and role of natural attenuation processes to manage this risk, developing technical basis for mitigation design and its implementation, and finally developing technical approach for remedy performance assessments. So as I summarized earlier, PNNL stewards a wide range of capabilities applicable for site characterization activities, and we develop an integrated approach for characterization for complex sites, utilizing the analytical capabilities specifically selected and needed to address specific data gaps at a site. So in general, an integrated site characterization approach focuses on understanding the contaminant biogeochemistry and its speciation in the subsurface. And this is an important element of characterization since it determines the contaminant transport, toxicology, and ultimate disposition aspects. And our approach also focuses on identification and characterization of site-specific attenuation mechanisms and controlling features and processes for contaminant transport. So we typically develop a tiered approach for site characterization, where we initially focus on bulk characteristics of the contaminant distribution and sediment, looking at things like total aqueous and solid phase concentrations at the site and some general physical properties of the sediments. Then with selected sediments and for selected locations, we move toward more detailed characterization of contaminants, particularly solid phase association at various scales and its transport parameters as needed at the site. This is sort of a busy slide, but here are some examples of some analytical techniques that we use to develop this integrated characterization approach. And I'll just very briefly walk through these to give you a sense of what we mean by these different capabilities and their integrated nature. We start with, again, analyzing the contaminant sediment, the core samples, as you see in this picture in the middle, for bulk properties and contaminant concentrations. And then they move towards techniques like sequential extractions, which would be contaminant specific. And these would be for understanding contaminant association with different sediment fractions. So we would be looking at different fractions from most mobile to immobile for the contaminants in the solid phase. So we also have a range of capabilities to interrogate sediment properties in order to better understand how sediments impact the transport behavior of contaminants in the subsurface. For example, based on the X-ray microtomography imaging that you see as XMT in the slide, we select certain representative cores for laboratory hydraulic testing. And we use techniques like multi-step outflow experiments to provide direct input into freight and transport models. So for contaminant transport behavior, which is usually in our tier 2 or later phases of the characterization approach, we conduct batch or column leaching studies. And here you see a picture of our some of our column experiments at the bottom of the slide, with a typical output from a flow leaching study for uranium. And these are these are conducted to provide transport parameters for these contaminants. And where we need at a site or specific location, we also rely on micron scale investigations using techniques like scanning electron microscope and energy dispersive spectroscopy, SEM, and EDS to gain information on morphology of individual grains and their chemical composition. So there are some other techniques we utilize like X-ray absorption spectroscopy, veins, to study interfacial reactions. So through a site-specific integrated approach, we interpret all of these results in terms of source term chemistry, geochemical interactions, including co-mingled contaminants, contaminant mobility and attenuation, and biologically driven transformations if it is applicable for the contaminant. And we use this information to evaluate potential remedy approaches at a given site. So these few slides provided a high-level summary of our site characterization capabilities in support of QCSM development. And my colleague, Vicky Freedman, will provide more information about these capabilities with a specific example. Vicky?
Sorry, I was having problems unmuting my video. We can do complex site remediation, but we can't handle Zoom calls, right? Okay, so can you hear me okay? Okay. So today I'm gonna talk through an example of some of the technical support we provided to DOE associated with radioiodine in groundwater. And what you see at the right-hand side of this slide is a map of the Hanford site, and it's showing you the footprints of sort of two distinct radioiodine plumes in the subsurface. The one that is at the left is the one that's the focus of today's talk. And the operable unit that this is focused on is called the 200 UP1 operable unit at the Hanford site. So back in 2012, when a feasibility study was conducted for this particular operable unit, it showed that there were no existing treatment technologies that were available that could achieve the federal drinking water standard of 1 picocurie per liter for radioiodine. And I will note that that is a very low drinking water standard. It's the lowest drinking water standard of all radionuclides in the Federal Register. So because there were no existing treatment technologies, there was a remedy that was put into place to hydraulically control that plume from moving from its current position eastwards toward the Columbia River. And then as part of the interim record of decision, that ROD dictated that 3 elements be investigated. One was to look at closing conceptual model data gaps for radioiodine. A second was to investigate whether or not there were existing remedies that could be adapted for radioiodine treatment, and that was looking at both in situ and ex situ approaches. And then simultaneously look at whether or not an applicable or relevant and appropriate requirements or an ARAR technical and practicability waiver, also known as a TI waiver, would be appropriate for this site. So we did all 3 of these elements concurrently. So just a little bit of background on the 200 UP1 iodine plume. It is that smaller one that exists to the west at the Hanford site within the Central Plateau. It's a pretty dilute plume. Currently, groundwater concentrations for radioiodine are right at that 1 picocurie per liter concentration, and they go up to about 23 picocuries per liter. And we are seeing some steadily declining sort of peak concentrations within that plume that's shown at the right hand side of this slide. The plume is fairly large, certainly not as large as the one that exists to the east, but it's about 3 kilometers squared. It spans about 1.2 kilometers wide, and it's fairly thick, about 45 meters thick. And that's important when you're considering, completing wells to treat the entire thickness of that plume. And then, the plume, for the most part, looks detached, that there is no continuing source. And the other really important aspect to provide some context in terms of really understanding quantitatively what's going on with the iodine conceptual model at the Hanford site is that iodine 127 exists in very large quantities relative to iodine 129. There was a resin analysis that was done on the Purolite resin that's currently being used to treat technetium in the 200 West pump and treat system. And it also captured iodide, which is a species of iodine. And it showed that the ratio of I-127 to I-129 was about 1,000 to 1. And of course, that's important because any kind of technology that you would apply wouldn't be able to distinguish between, stable iodine and radioiodine. So your treatment technologies would need to be very efficient in order to capture all of the iodine 129 that would need to be treated at the site. So as part of the quantitative conceptual site model that we formulated, through some laboratory investigations, and we're focused on identifying what the species were in the subsurface. So iodide, iodate, and organoiodine are the prevalent aqueous species in groundwater, but iodate predominates, and that was contrary to what we initially thought. We thought that iodide would have predominated in the subsurface. Traditionally, our models have used KDs for transport associated with iodide and not iodate, but iodate actually happens to interact a little bit more with sediments than iodide does. For example, it interacts with calcite. There's some co-precipitation that occurs under just sort of natural conditions. And it also interacts with iron oxides and co-precipitates and also sorbs. And then we also identified kind of a nominal capacity for sediments to volatilize iodine in the subsurface. So any iodine, for example, if there's microbial activity in the groundwater, there could be a small amount of radioiodine that might volatilize, but it was really very small. And so that wasn't considered a very important component of the conceptual site models in terms of it being able to naturally attenuate radioiodine in the subsurface. And then just a note that we did produce a manuscript. We co-authored this with Savannah River. And the figure that you see to the left shows where sort of all the different forms of iodine and species that can form. But one of the reasons we wanted to look at the conceptual models between Savannah River and Hanford sites was because there were significant differences in climate, for example, and sort of the redox conditions at the sites that mean that iodate is predominant at the Hanford site, depending on where you are at Savannah River, close to the source term, or if there are wetlands at the site. They're seeing that it's iodide and organoiodine that are predominant. And again, all this information is really important in terms of identifying potential technologies for treating iodine in the subsurface. Okay, so in terms of the technology identification approach that we put into place, the first thing we did was we performed a literature review to identify any promising to be out applied subsurface contaminants that could be modified for use with radioiodine in the subsurface. From there was a down select a process, and we identified some scoping tests where further evaluation was needed for performing some extended and scale-up tests to see whether or not we could move into that next phase. And Inci had talked a little bit about some of the capabilities that we have in transitioning technologies from sort of, the laboratory scale to the bench scale to the field scale. And that was the intent of our technology evaluation. And then, of course, one of the considerations we had to keep in mind, knowing that there's lots of stable iodine in the subsurface, but we needed to meet that maximum contaminant level of 1 picocurie per liter at the site. Oops. And so we divided the technology approaches into 3 categories. There was in situ sequestration approach, where we just immobilize the iodine in situ. We also looked at mobilizing iodine. And of course, if you're going to mobilize it, you have to have a way to capture it within a pump and treat system. And so the idea was to enhance that efficiency. And then a third category was really just to identify an approach that could be used within the pump and treat system itself. We did have a look at whether or not microbial-based technologies should be included, but in that literature review, in that first step, we ended up eliminating microbial-based technologies because, one, they seem to be not a very significant process in the conceptual model and that quantitative conceptual site model evaluation. And then there were already some operational difficulties associated with just a biological system that was causing a lot of biofouling at the pump and treat facility. Okay, so on this slide, I'm just summarizing some of the in situ sequestration and technology screening results. We are aware that calcite co-precipitates with iodate, and that's the predominant iodine species in the subsurface. but we found through our column experiments that if we were to promote calcite precipitation and subsurface, that only resulted at best sort of a 70% removal of iodate only. And because we have so much stable iodine, that was not efficient enough, and it was eliminated as a potential technology approach for treating radioiodine. We also looked at iron oxide sorption as well as co-precipitation. The nice thing about the iron oxide was that it could not only remove iodate with a pretty high efficiency, but it also removed iodide. And the relative percentage of iodate to iodide at Hanford is probably have about 70 to 75% of iodate, only about 5% of iodide, and the rest are sort of organo iodine species. This one again was eliminated because of the reversibility of the reactions, and so it was not considered one that would go forward into treatability testing. The other consideration associated with iron oxide as well was not only that it was reversible, but that it also, because of the size of the plume, and the inability to treat or to drill wells within the core of the plume. And one of the things I forgot to mention on a previous slide is that the Erdif landfill disposal facility is located right above its core, and its expansion zone is located right along the iodine trajectory path. And so that created just some sort of implementation problems associated with drilling wells within that area. And then dithionite, we saw that you could get up to 4 times more and 3 times faster leaching of iodine. That's this what's shown in this graph here at the bottom. But again, you have those operational difficulties associated with drilling and pumping and treating that water. But there were also some foulants that it also released, like sulfate, and it was not a practical technology to look at. And then we also looked at some commercial resins as well. And even though some of the batch testing demonstrated a reasonable iodate retention, when we transitioned into column studies, those resins showed that they had a fairly low capacity for iodate retention, and even had some negative interactions with groundwater constituents. So those were all eliminated from as potential technologies for treatment of radioiodine at 200 UP1. And so the technology evaluation conclusions were that all the practicability of the candidate technologies was low. It's driven by where the core of the plume is located, the site and contaminant properties, and really that sort of high efficiency that you need for the technologies to be able to treat sort of the relatively small percentage of radioiodine relative to stable iodine. And we worked through with the EPA sort of an EIC, effectiveness of implementability cost evaluation, for the technology maturity following the CERCLA process. But all of this information, so the conceptual site model information supported the technology evaluation, and the technology evaluation and the conceptual site model supported the consideration of a TI waiver. And that's sort of the next piece that I'll talk about. Some of the work that we did associated with the TI waiver support was creating, based on an EPA memo that was published in 2016, a checklist associated with the kinds of information that's needed to support the request for a TI waiver from the EPA. And so part of that information was just gathering conceptual site model information that we had identified through our QCSM development process. We also had the information on the technology evaluation. And then there was also this third element where we needed to be able to look at sort of the restoration timeframe analysis. And so part of that, we support the site contractor. The site contractor is actually responsible for all the decision-making modeling that's performed at the Hanford site. But we wanted to be able to support that restoration timeframe analysis. And so in part what we did was we did some simulations of some of the column experiments that we were able to look at that would provide us transport information, kind of the KD information needed for doing sort of the more field scale modeling. And so what you're seeing at right is data that was collected from experiments where you had sediments that in the absence of sort of ferrihydrate amended sediments, and then in the bottom here, that's where the ferrihydrate was amended. And what we found is that the sorption behavior had an impact on how the plume behaves. What we saw that if you've got a really slow moving plume like this iodine, when it first comes in contact with fresh sediment, you see a lot of initial absorption. The sorption sites get filled, then the plume moves and then stalls again when fresh sediment is encountered. And so we ended up identifying a Langmuir, a kinetic Langmuir isotherm approach, which included this sort of finite capacity for the sediments. And this is some of the knowledge that got transferred to the site contractor that's responsible for this modeling. And then we also did some field scale simulations where we considered coupled vadosone and groundwater system. And we used eStomp, which is one of the codes that you're able to use at Hanford for decision making. I stated previously that this was a detached plume, but we wanted to historically simulate all the iodine sources and make sure that there weren't any slugs of iodine that were going to come through at a later date. And we were able to confirm that with some of this coupled vadosone and groundwater modeling. So that was some information that we were able to transfer to the site contractor. One of the another sort of final element in this sort of iodine conceptual model support story is that even though we've identified that there's no technologies available and that we've got and that there is no continuing sources for iodine, there still is an ongoing need for measuring iodine in the subsurface, and especially I'm sort of starting and ending with the same graphic. There is a larger, more dilute plume that's located in the east area of the Hanford site. But we ended up developing kind of a rapid sort of technique for simultaneously measuring iodine speciation in the subsurface. So you have both iodide and iodate. And of course, that's important because one, if there is a technology that needs to be applied, that is something that is needed, but it also provides information, continuing information to support the site contractor in the simulation modeling that they do to help make decisions at the Hanford site. This method has 2 to 5 times lower detection limits than standard methods. This 1 picocurie per liter is equivalent to about 5.6 nanograms per liter. So pretty small. For iodine 129, we can measure down to 1.8 nanograms per liter for iodate and 2.62 nanograms per liter for iodine. And so I think, oops, I'm having a hard time controlling, but I think this is sort of my final slide. I just wanted to talk through sort of all the different elements that were used to provide this technical support to DOE. There's, biogeochemical characterization in terms of identifying the predominant species and at the 200 UP1 operable unit. There was technical support in terms of identifying the technologies that could be used. We used simulation to support our knowledge base and make sure that we understood those processes. Through the TI waiver support, there was, you have to be knowledgeable about the regulatory requirements. And And then we didn't get to a point where we advanced into treatability testing, but we did get up to the point where we were doing some initial screening. But all of these elements need to combine together. There's certainly the core rests on the quantitative conceptual site model development. It takes an interdisciplinary team. And then throughout this entire process, we tightly integrated with our sponsor, with the regulators, and all along the way, transitioning the findings and communicating the results. And so with that, I'm going to turn this back over to Melissa, who's going to, I think, facilitate the question and answer period.
Yes, thank you, Vicky and Inci and Tom. We have about 40 people online with us today, and we have a couple questions that came in. The first one is for Inci. What drives the costs associated with characterization activities at complex sites?
Thanks, Melissa. So the costs, I guess one of the primary elements of cost is the borehole characterization, borehole numbers, and the construction. So majority of the site characterization information is derived from these core samples that I showed the picture when I was talking. And especially for largely distributed contaminants in the subsurface, you can imagine that the numbers of boreholes that we would need in order to understand the actual distribution of the contaminant could get very large very quickly. So that would drive the cost significantly at a site. However, recently we are trying to use methods like geophysics in order to guide the borehole locations for site characterization. So that would, with those techniques, we can very quickly and easily look at the subsurface and identify the contaminant distribution and in important aspects of it in order to better locate these boreholes. And that helps streamlining the process and reducing the overall costs at the site.
Okay, thank you very much. There's another question. I'm not sure if it's Inci again or if it's Vicky. What are the most important parameters, both sediment specific and contaminant specific, to measure for contaminant transport and risk assessment models?
I can start and maybe we can add. I say for the sediments, obviously the transport properties are important. So hydraulic conductivity is a parameter that would be very significant in order to understand how sediments would affect the mobility of the contaminant. And for the contaminants, its speciation is very important in order to understand the mobility of it and biogeochemistry of the contaminant. And I'm sure Vicky will probably expand on that too.
I think I would just add to that certainly half-lives and how long a contaminant is going to stick around for, and whether or not you would expect the contaminant to, its concentration to diminish before it reaches its receptors is something that's fairly important. And I think too, the presence of other contaminants that might affect, that contaminant transport behavior.
Okay. The next question is definitely for Vicky. If there aren't any technologies available to treat I-129, why not implement MNA instead of a TI waiver? Was MNA considered as a potential treatment?
So yes, MNA was considered as a potential treatment. It wasn't included in the technology evaluation because it was identified early on as part of the quantitative conceptual site model development. MNA generally needs to be completed within a reasonable time frame. There's kind of this general consensus out there that a reasonable time frame is maybe about 50 years. But the reasonable time frame for radioiodine would be much longer on sort of century scale. And so what the TI waiver would afford in terms of management is that you wouldn't necessarily have to monitor within the core of the plume, because that core of the plume would be exempt from having to meet the drinking water standard, cleanup standard within that core. And so that would be the advantage of using a TI waiver. But of course, the TI waiver gets revisited every 5 years. So it's not permanent, but it does allow a little bit of flexibility.
All right. So this next question, I believe, is for Inci. It's a little technical, so I'm going to try to make sure I get all the words right. Experiments have shown high efficiency of iodide methylation and volatilization by UP1 sediments across a range of site-relevant concentrations. Iodide is the more abundant chemical species. Still, why wouldn't processes that affect the iodide mass so significantly not be considered in the QCSM and projected into the future?
I can answer that, Inci.
You got that one, Vicky? Okay.
Yeah, I'll save you, Inci. I think what those experiments applied heat. And so that was why it wasn't considered a significant process, because and also you have to kind of consider what's happening under ambient conditions and what's happening in groundwater. And so the thought was that, and I think there was some modeling that was done as well that showed that it was a process that was that could be occurring, but that it just wouldn't be significant relative, for example, some of the iodate and iodide absorption to sediments and interaction with, both ferrihydrate and calcite.
All right, we have another question that just came in. What distinguishes a QCSM from what might be considered a more traditional CSM?
Do you want to take that one, Inci, or you want me to do it?
You can go ahead and
I can, as I said, I can read it again.
No, I'm good. For me, when I consider a conceptual site model, for me without the Q, I think is more focused on kind of defining the current state. But for me, the QCSM kind of says, this is how we got there. This is why the current state exists as it is. And then it also gives you some additional information that can be used, for example, in identifying, potential technologies.
Right. And I can add to that. I think the quantitative nature of CSM is associated with understanding the time frames for the processes that are occurring, that are observed in the subsurface. So this would add, the Q would add the time frame information to decision making. And as Vicky identified, the CSM is more qualitatively describing the site information, as opposed to adding more actionable information like the rates and other components that would be associated with the time frames.
So we don't have any additional questions, but Tom, you didn't get any questions. You're off the hook. Do you have any wrap-up comments or anything that you'd like to elaborate on?
As soon as I unmute, yeah, I just want to thank everybody for participating. I know that one of the drivers for this specific discussion topic today was a request from audience from some of the early seminars on the Remplex series that wanted to kind of understand the characterization or the capabilities, the core capabilities that are important to complex site remediation. So I hope those of you who were able to participate got a flavor for that. Obviously, we touched on really just an overview of the broad set and a more specific look at some focused on the QCSM. I think if you're interested in this topic, we'll certainly work to mix in some future seminar topics that get at some of those other core capabilities and how they're, with some additional examples of how they're being applied, and also look forward to us starting to bring in some of our collaborators in this seminar series in the future, so that you can not only get the perspective from our PNNL experts, but from some of our partners and other experts in the field that have maybe some other perspectives and other complex sites that they're working on that will help round this out. So thanks everybody, and sure appreciate everyone's interest in the topic and questions.
Absolutely. And thank you everyone for joining our seminar today. A reminder that our seminar video and the presentation slides will be archived on the Remplex website. And our next seminar is December 1st. I just posted the flyer in the chat box. It's on calcium carbonate minerals as scavengers for contaminants, both metals and radionuclides. So that's December 1st. We have a couple ways you can keep in touch with Remplex and our activities, and to reach out to our team to look for collaboration opportunities. We're always looking for more partners. So we have a newsletter. So if you want the newsletter, you can either put it in the chat box or send an email to remplex@pnnl.gov. And we also have a LinkedIn group, and we'd love to see you joining us on LinkedIn as well. And with that, thank you very much to Tom, Inci, and Vicky, and thank you for those of you who spent your lunch hour with us, and we'll see you on December 1st.